So perhaps when you think about genomics and ART, the first thing that comes to mind naturally is PGT. Make no mistake about it, successful identification of euploid embryos absolutely is critical to IVF success. And this is also an area that Cooper Surgical spends significant effort innovating and developing out solutions. Our PGTAI platform is summarized here, and you can find more information at our website or our YouTube channel. But for the purpose of today's talk, we're going to be starting with the assumption that we've identified euploid embryos and then discussing additional factors that influence IVF success. As a quick refresher, the endometrium is the inner lining of the mammalian uterus, which is a dynamic organ that undergoes cyclical changes as part of the menstrual cycle. First, we're driven predominantly by estrogen, which drives proliferation of the luminal epithelium and endometrial glands. And then following ovulation, really we shift to more of a progesterone-mediated biology. Now, in contrast to some other animals in which the embryo is really the key driver of pregnancy outcomes, the endometrium plays a really important role in human reproduction. The endometrium actually has remarkably sensitive ways of acting almost as a sensor to evaluate the putative implanting embryo. If everything goes well, implantation triggers a proliferative and differentiation response of the endometrium known as decidualization, which nurtures and supports the developing embryo and eventually contributes to the placenta. However, if the developmental competency or viability of the embryo is lacking, the endometrium can sense this and it will trigger menstruation so that it can begin moving on to the next cycle. Taking a step back from some of the complex molecular networks, the ultimate goal and objective of the endometrium is to support an implanting embryo. As we discussed earlier, the endometrium is a very dynamic organ and it's constantly undergoing changes. In fact, throughout the 4-week menstrual cycle, there are really only a few days in which the endometrium is receptive or willing to receive an implanting embryo, and this phase is known as the window of implantation. In natural cycles, the window of implantation occurs approximately 7 days after the LH surge, in most women, that is. In hormone replacement or controlled cycles, typically progesterone is administered, and the idealized window of receptivity is considered to be 5 days after progesterone administration. The concept of the window of implantation or restricted receptive time really originated and was first hypothesized in the 1950s when endometrial dating really first came to be based on histological examination, such as I showed earlier in the presentation. The presence of a narrow receptive window surrounded by refractory periods later was shown to be clinically relevant in the 1990s, and for decades it was thought that all women would have similar underlying physiology. As we all know now, following the successful Human Genome Project, science saw a tremendous explosion in capabilities. Many tools were subsequently developed, and with those tools we began to really gain an understanding of the molecular mediators of our physiology. With that increased understanding, we began to define many of the molecular networks and interactions that I've described previously. Scientists then began to use these newly developed tools to investigate some of the differences in our physiology and the molecular mediators in various different conditions. Without any surprise, the uterus, such a dynamic organ, really offered a lot to discover. Starting broad and drilling down, scientists first really focused in on molecular differences and changes in the endometrium throughout the menstrual cycle. In the graphic here, that's demonstrated on the x-axis of time. So starting on the left, we have a dotted line signifying tissue that could potentially represent menstrual tissue, and we continue to the right to have early proliferative, late proliferative, early secretory, mid-secretory, and late secretory, including the window of implantation right at the beginning of mid-secretory phase. Moreover, as our tools got better, we were able to begin characterizing and profiling various different cell types. So in this graphic, you can see multiple cell types with a key on the right, and on the top and the bottom of the figure, what we're actually showing here is gene expression profiles of several samples of epithelial cells and stromal cells throughout these cycles. Now, we won't key in on what the specifics are here, but each vertical little stripe is a single gene. And as you can see how that gene, based on the color intensity with the code on the bottom left, changes in the respective compartments and in the respective menstrual phase. Though you may not be able to make perfect sense of it, hopefully your eyes can pick out some patterns and some differences throughout the illustration. Eventually, what scientists were able to do was to leverage statistical classifiers and various different algorithms to establish a gene signature that reflected any given cell type or any given phase of the menstrual cycle. Once these signatures were established, we could then begin comparing patients who presented different phenotypes and prognoses and compare their signatures to one another. Using the previously discussed techniques and signatures is when we eventually came to discover that not all patients are the same. And so in the middle, we have the typical scenario, if you recall, 5 days after progesterone administration or 7 days after the LH peak, we would expect the endometrium to be receptive as well as to have a receptive signature. If we were to examine the signature of that patient earlier, say 2 or 3 days after progesterone administration, or perhaps later, say 7 days after progesterone administration, we would expect to find signatures that perhaps reflected more the early secretory phase or the late secretory phase, respectively. However, in some patients, we didn't always see this. And so on the top, you have what we call as pre-receptive or delayed window. And in this case, if you perform the biopsy at 5 days after progesterone administration, when you expected to see a receptive signature, you would actually see a signature that indicated you hadn't reached the receptive window yet. And in fact, if you biopsied a subsequent day, you would find a receptive signature later than you expected to find. In the exact opposite scenario, known as post-receptive, if you biopsy that patient at 5 days after progesterone administration, you would find, in fact, that the gene signature had advanced and had passed the window of receptivity. In patients with these kinetics and physiology, you would actually have to perform a biopsy at an earlier time point to identify a signature that suggests that the endometrium was receptive. Here we demonstrate the translation of these gene signatures to clinical practice. So again, recall we may have a few patients, some who are receptive, pre-receptive, or post-receptive. When we conduct molecular analysis, as demonstrated here below, we can look at the gene expression and establish gene signatures that reflect those physiological states. These signatures can be transformed, statistically speaking, into a two-dimensional space where we see unique populations. In green here, we have a group of patients who are populating close together that we know to be pre-receptive. In purple in the middle, again, we can see a distribution of patients who are demonstrating a receptive signature. And then over here, we have a group of patients who are demonstrating post-receptive gene signatures. And then finally, a fourth class of non-receptive signatures. When we look at the gene signature derived from patient 1, we see that this sample clusters right in the middle of the pre-receptive group, and therefore this patient must be pre-receptive. In contrast, when we look at patient 2, we see it mapping and identifying in the post-receptive group, and finally here patient 3 is mapping in the middle of the receptive group. If you'll recall from the previous slide, in addition to using the receptivity terminology, we also discussed delayed and advanced windows. And so, of course, patient 3 is our easiest patient who has a receptive window, which means that her endometrium is prepared to receive the embryo at exactly the time that we would expect, and transfer should be performed at that same time. In contrast, where things get a little bit more difficult, we have patient 1 who is pre-receptive or has a delayed window. This means that as far as physiology and gene signatures indicate, her endometrium hasn't advanced to the window of implantation yet, and therefore transfer would be better suited if it were delayed. Finally, we have patient 2 who is post-receptive or has an advanced window. In this case, the gene signature indicates that her window has already passed, and the ideal time for a precision transfer would be 1 day earlier. Based on these principles, Cooper Surgical has identified gene signatures that reflect the various receptivity states, and we are proud to present ER-peak endometrial receptivity testing. This test is intended to be performed on endometrial biopsies that were obtained under the same timing and hormone paradigm that you would utilize for embryo transfer. While some of the concepts related to receptivity, gene signatures, molecular mediators, etc. are pretty complicated, we're happy to present the results in a simple and easy to understand graphic shown here. If the patient tests receptive, then we've identified the ideal time to transfer an embryo under the exact same conditions in a subsequent cycle. If that is unfortunately not the case, the subsequent cycle, the embryo can be transferred with a shifted window, either 1 day early or 1 day late, or a second biopsy can be taken for confirmatory testing to confirm that the shifted time period reflects a receptive status. So far, causes of a shifted window still need to be investigated. However, we do know that patients with recurrent implantation failure, as well as patients with higher BMI, exhibit an increased incidence in a shifted window. These patients are often considered ideal candidates for receptivity testing. Additionally, some patients who have only a single embryo available or a single euploid embryo available also may find receptivity and window testing very important. In approximately 2 years of testing, Cooper Surgical has observed that approximately 60%, the majority of patients, are in fact receptive. However, approximately 2 in 5 patients presenting and requesting ER-peak testing are in fact not in the receptive window. Of those patients, we do see approximately 27% of them who are pre-receptive, while approximately 7% of them display post-receptive signatures. It's important to note that within these data, we have not stratified out any patients based on their indication. Identifying altered physiology is one thing, it's great, but what about implementing intervention and clinical practice? Here we have clinical data from 2 very compelling and interesting stories. On the left, we have patients who, in a retrospective study, were identified to have a shifted window. In the dark blue bars, this was their first cycle under the standard time frame, and we can see relatively poor outcomes. Once the shifted window was discovered, a subsequent embryo was transferred in the precision embryo transfer period, in other words, in accordance to the signature observed, and we can see marked improvements in implantation rate, clinical pregnancy rates, and ongoing pregnancy rates. It should be noted that in the original standard embryo transfer, not all pregnancies were followed to full ongoing pregnancy, and that's why that data is missing. The study on the right is separate patients, in contrast to the patient-controlled study on the left. And in this population, we have recurrent implantation failure patients who have undergone standard embryo transfer during typical timeframes, or those who have had receptivity testing and then undergone a precision embryo transfer. And again, we see significant increases in implantation rate, ongoing pregnancy rates, and live birth rates.